Good afternoon, everybody. It's really a treat to be here and very much looking forward to the science for the rest of the day here. So as mentioned, let's see if we can get going here. There we go. Okay, so as mentioned, I'm going to tell you a little bit about the sort of broad themes of our research program at Simon Fraser University, and then we're going to take a deep dive into one of the technologies that we've developed recently, which is in the area of sort of throughput for untargeted metabolomics acquisition. So as mentioned, my lab is really interested in the study of natural products, so we're interested in molecules found in the natural world, and really at the broadest sense, the lab is interested in these two key questions. We are interested in contributing to methods that might help us understand the chemical diversity of the natural world, and then also how we might apply this chemistry to problems in human health. And obviously, both of those are enormously challenging and complicated problems. We don't intend to solve them on our own. This is part of a big sort of field-wide effort. But I think we're really interested in questions that surround the biosynthetic capacity in nature, like what might we expect to find in the natural world, what has been found already, and where are the opportunities for new applications. I think that really kind of drives a lot of the science that we do. Anyone who's ever done natural products will perhaps recognize this cartoon. So this is a general schematic of how one goes about doing natural products-based science. So this hasn't really changed all that much since the earlier part of the 20th century. The discovery of penicillin and erythromycin and others followed this same workflow. So in very general terms, you start by going into the field and you collect a set of organisms. These might be microorganisms or plants or marine invertebrates. And you then convert those organisms into a set of extract libraries, which you have now in modern times arrayed in typical well plate format most commonly. Those libraries then get analyzed both with some kind of chemical analytics, most commonly LC-MS and LC-MS-MS. And then they also get subjected to some sort of biological evaluation. And this might be antimicrobial testing or testing against stable cancer cell lines, or it might be chemical ecology and applications in nature, but there'll be some kind of biological evaluation. And then those two streams of information that come from these two different types of experiments get integrated in some way. And in the olden days, this was with Excel, and in modern times, this is with more sophisticated technologies. And then what follows afterwards, so the goal of the integration is to identify which molecules are putatively bioactive agents, and then you use databases and other computational tools to deprioritize molecules that have low value and invest your valuable time in isolating and identifying those things with the highest return. We estimate in our lab that we still spend about 80% of project time on isolating and identifying molecules. That step is really slow. And so decision-making at this stage is really important. You have to be able to make good decisions. So over the last 5 years or so, the lab has become really interested in sort of informatics technologies and how we might contribute to this workflow, and in particular, identifying bottlenecks in the discovery process which might benefit from algorithmic solutions. So we've developed a number of tools and technologies which aim to address specific problems in this pipeline. I'll just tell you what they are. I'm not going to tell you about all of these today, don't worry. But I will tell you just briefly what they are so that you have a flavor for what the lab is interested in. So the first one on the left here, multiplex MS. This is the tool I'm going to describe in the talk today. This is a tool designed to improve acquisition throughput. MS2Analyte is a platform-independent data processing pipeline that's designed from the ground up to handle natural product samples. NPAnalyst is a web server for doing data integration between untargeted metabolomics and screening data sets. The NPAtlas we'll come to in a minute. This is a database of all known microbial natural products. SnapMS is a tool which uses the chemical diversity found in this database for de novo identification of natural products. And then NPMRD on the right here, this is a consortium project to build a database for NMR data for natural products. And so you can sort of see how each of these might sort of address some outstanding challenge in that general field. But the area where we're going to focus most of our attention today is in multiplex MS. So if we think about how natural products science is prosecuted, it's gone over the last 30 years from being a relatively the linear process where people would study samples a few at a time, and each student might study one sample at a time, through to a situation where now, with the advancement of liquid handling robotics and lab automation and informatics tools, it's possible to study those samples in sets that are much larger than just a few at a time. And really, we'd like to study them thousands or tens of thousands at a time. So what I'm showing you here is a summary of a project we did where we took a set of strains from our marine bacterial library. So another hat that I wear is that I'm a scientific diver, and so we go in the environment, we go diving and collect samples and collect isolates from them. So these isolates come from things we've collected up and down the west coast of the US. We turned them into cultures from which we made extracts, and then we used a combi flash to do a very simplistic polarity-based 6-step fractionation, and we made a library of just under 1,000 samples. We then took those samples and we screened them in 15 different antimicrobial assays for a total of whatever that is, a little bit less than 14,000 data points. And that's a very fast experiment to do. Liquid handling robotics for screening is now extremely well established. It's not that difficult to do assays on that scale. That whole thing took less than a week. But we also analyzed those same samples in triplicate using our SYNAPT G2SI. And that takes a really long time. Because if you want 3 replicates of every sample, and there are almost 1,000 samples, then you have to run almost 3,000 runs. And if your duty cycle is 8 minutes or 7 minutes or whatever it is, it's going to take really, really a long time. So we did it, but it was a chore. And so this is an obvious bottleneck for us, that there's this really dramatic disconnect in the order of magnitude of things you can analyze in the one modality versus the other. And so for us, that's something that we really wanted to try and address. So this is the problem we're trying to fix. So it was mentioned before that we run a database called the Natural Products Atlas. So this is a database that we run with a whole suite of volunteers who help us to do the curation. It purports to be a database that contains every microbial natural product that's ever been published. Now, to really be sure that you got that right, you would have to read every publication that had ever been written, which is obviously quite difficult. So it's a constant effort on our part to try and keep this database up to date. But the database is, like all the tools we build, it's open access, it's completely free, it has a completely unlimited license on it, commercial or non-commercial. It works on any platform. It's a web-based system. If you're a programmer, it has a full-blown API on the background. And it also, you probably can't read it, but here there's a download section where you can download the entire database in a bunch of different formats. And we built the database in large part because at the time we started, there really wasn't a repository which contained all that information from the literature. But once you've built a database like that, there are lots of interesting things that you can do with it. And so one of the things that we did with it quite early on is we just asked, well, what's the scope and scale of diversity of chemistry in this database? So if we do the simplest possible thing that you could think of doing, we took all the molecules in the database, we took all the formulae from those molecules, and we just counted the number of times they showed up. So this plot here on the x-axis, it's ordered by number of carbons, number of hydrogens, and then all of the other bits of the formula are ordered. It's ordered alphabetically in the way that you would normally write formulae in chemistry. And then on the x-axis, of course, is the frequency count. Oops, got the wrong button here. And what you can see from looking at the data from the entire database, so the database contains about 37,000 compounds at the moment. And what you see is that there are a handful of formulae that are really prevalent. These are C15 formulae. Those of you who know about biosynthesis in natural products might not be surprised to see that C15 compounds they show up a lot. But if we zoom in a little bit more closely, so if we pick 20 carbons as a representative example and zoom in and look more closely at those data, 2 things stand out. So the first thing that stood out to me is that I thought something had gone wrong with the algorithm that we had used to count the formulae, because these seem oddly, they have an odd periodicity to them. It looks like a very strange plot. But when you look more closely at what those high peaks are, you find that these are all the formulae that only contain carbon, hydrogen, and oxygen. So what this tells you is that in natural product science, if your formula has anything other than carbon, hydrogen, and oxygen in it, then the number of known matches in the database is really quite low. And that actually the diversity of components in natural product samples is quite high and quite broad. And that suggests that if there isn't a lot of high repetition, and if there's a lot of diversity to the formulae that are out there, that maybe we can take advantage of the latest generations of mass spectrometry tools and technologies to speed up the acquisition by running samples not one at a time, but in large pools where we use the resolving power of the instrument to resolve those pools, and we use computational algorithms to unpick those data back to individual sample lists for individual samples. It's one of the only ways one can think of to speed up that process in a significant way. So this was the original premise, this was the prospect, this is what we thought we would try out. And so we designed a method for data analysis where you take your set of samples and you assemble them in theoretical grids. So this is a cartoon example. So here we've got 16 samples arrayed in a 4x4 grid. And then you make pools by pooling across each of the rows, and you make a similar set of pools by pooling each of the columns. And you only run the row and column pools. Now, obviously, in this toy example, you don't get much of a time saving for this. But you can imagine if you made the grids much bigger, you would start to get a quite significant time saving. So bear with me with the small cartoon grid here. So what we actually do, for reasons I'll explain on the next slide, is we don't run just a single grid. We take the same set of samples, and we array them differentially in two different grids. So we make two sets of pools of columns, two sets of pools of rows, and then this gives us an inherent four-way replicate analysis. And you can imagine, if you take just the top grid here, you can imagine that if you see a given feature in row 3 and also in column 3, then you know that it must have come only from this sample that's at the intersect between the row and the column. And you can use that premise to unpick the row and column data and reassemble those data back into feature lists for individual samples. So this sounds quite attractive. It sounds like a fun prospect. But those of you who are keeping score might recognize that there's a problem with this. And the problem with this is that if you take just a single grid and you find your signal in one row and one column, then determining its origin is really easy. But if you have a scenario where you have the same feature in two different samples, then you're going to get signal from this row and this column because it's present in sample 9, and this row and this column because it's in sample 17. And the problem with that, if you look at this observed grid, is that you can no longer differentiate between a scenario where the feature is in 9 and 17 and a scenario where it's in 7 and 19, or some combination of those. So you get in a mess really quickly. It gets really difficult to deconvolute. So we learned two things from this. One is that single grids are not going to be very useful. And the other one is that this will work best for features which are generally rare in the data set, which fortunately for natural products applications is usually true. So it's for this reason that we take the dual grid approach. Because, as we'll see, if I just back up one here. So you can imagine that if you find a given feature in this row and this column in the first grid, but also in this row and this column in the second grid, and you know the positions of the features in both grids, deconvoluting the data set becomes much more straightforward. So this is a much more precise and accurate way of doing that. And the way we rebuild those grids is using a sort of Rubik's Cube type approach. So you can see here on the top left here, we've got samples 1 through 25 just arrayed horizontally. And if you take this layout and you sequentially bump each row, each column up by one more, and then take the samples that are off the end of the grid and put them down in the bottom, and then you do the same thing horizontally, you can rearrange the second grid so that no two samples in the second grid, no two samples in any row or column in the first grid are in the same row and column in the second grid. So you get the maximum diversity of information from those two grids. So it's a bit like a Rubik's Cube, you can imagine, sort of twisting it in both ways and muddling it up. It's sort of the same idea. So you might think that this is a sort of cute idea, but it doesn't work very well in practice. And so one of the things that we did was to test whether or not we could still observe features that we would expect to observe as feature complexity, as sample complexity got higher. And to do this, we took a sample from our library that we knew contained, so here's a reference sample from our library. This is one of our extracts, and we knew it contained these two known natural products for which we had standards. So if you run the standards and you run EICs, you can see their traces on the top. And if you run just this sample on its own and you run the same EICs, you can see those same signals there. And then we prepared sets of mixtures, adding sequentially more and more and more other samples from our library, controlling the concentration so that the reference sample was at the same concentration in every one. And then we looked to see whether or not we could still observe the standards. And you can see that the signal shape and the intensity remains pretty constant throughout. There is a bit of an intensity drop-off in the last one when you get to 100, but 100 is a pretty big number, so we'll take 50. So if you do this with other samples in the set, you see very similar performance. So this is a second example. Here we're looking at 3 different molecules. So this is a different sample. It's got 3 other natural products whose identities we knew. And what I want you to just look at here are the peak shapes. So this is, the color coding tells you how many other samples were in the mix. And you can see that the peak shapes stay very clean and standardized. You see a little bit of an intensity drop once the number gets very, very high, up towards 100. So then the last thing we wanted to know is, well, how good is the recovery of this approach? Is it actually going to work for accelerating data acquisition? And for this, we took one of the samples in the library and we ran it 3 times. And then we took all the features that were present consistently in all 3 replicates as our gold standard reference set. And then we made the same set of mixtures as before, but now looking for all the features in that set, not just a single feature. And we asked what proportion of the gold standard features could you find if you added 1 more sample or 2 more samples or 3, et cetera, up to very large numbers. And what you find is that if the number of samples is not too high, below 15 or so, 20, then the recovery rate is very good. And then gradually, as the number of samples increases, the recovery rate starts to deteriorate. And we didn't really know why that was. And so the plot in the bottom right here are the same set of reference features, now organized by relative intensity. So the most intense feature is at the top, the least intense feature is at the bottom, and then the color coding tells you what the intensity was of that same feature as you add more and more samples. And so what you find is that, perhaps unsurprisingly, it's those low intensity features that are close to the limit of detection which become hard to find as the sampling complexity increases. We looked at a lot of these to see what they were, and a lot of times they're either small isotope peaks like M4, M5 isotope peaks, or they're minor adducts or fragments from molecules that have other MS features in the spectrum. So although you lose a little bit of information content, you don't actually lose much content about the compositional information. You just lose some of the fine scale detail about the isotope patterns when the samples get really complicated. OK, so in principle, if you run a 20 by 20 grid, then you get a 95% improvement in the acquisition speed. So the acquisition takes 5% the amount of time that it would have done if you ran them independently. But what we'd really like to do is use these kind of methods for really large experiments. So as a thought experiment, we asked, well, what happens if you want to run 500,000 samples on a chromatography-based system? So if you want replicates and you're going to run 500,000 samples and it's a 7.2-minute gradient, it's 27 years of machine time, which is pretty impractical, right? My team would be very unhappy if I proposed such an experiment. So that's obviously not going to help. And so this is just a theoretical plot which tells you how many samples you have to run as a function of the size of the grid that you make. And it's an inverse relationship. So you can see that you get a dramatic improvement in the number of samples you need to run goes down a lot if you can increase the grid size even a bit. It makes a huge difference at the lower ends. So if we go back and we look at so this is data from our SYNAPT. This was in-house in our own lab. And remember, this plot is showing you what proportion of the ground truth features in a given sample can you also find if you add more and more samples. So if you took arbitrarily 80% recovery as the recovery that you were willing to accept, then the maximum number of samples you could run is 22. But fortunately, we also were offered some early access to the Xevo MRT to see whether or not that would help us, and we expected that that additional resolving power and the speed of the detector would both have a positive influence. And so we did this exact same experiment. I'm not sure Waters was really expecting us to ask to run so many samples when they offered us a chance to use the prototype, but they were very good-natured and they let us do it. So here's the Xevo. You've seen this already. And the results are really amazing. So if we look at the recovery rate at 80% now, we can screen 60-something, 63 samples at a time, I think. You can overlay these two plots together. You can see the difference. And so this makes a huge difference, because if you want to run experiments really on scale, I mean, maybe 500,000 is an unrealistic objective, but perhaps you want to do 50,000. I think that that is a realistic objective. And I think that the difference in acquisition rate will be really dramatic using the additional capability of new tools like this. And of course, in addition to this high recovery, you also get beautiful looking spectra. So these are we just picked these out of one of our samples. So we know quite a lot of natural products that come from some of these materials. So this is just one natural product we come across quite a bit. And so this is the SYNAPT data on the bottom, which is beautiful. This is the MRT data above, which is just like the next level. It's really, really something else. So if you're interested in the MRT, sorry, not the MRT. If you're interested in multiplex MS, as well as the theory, we also built an app so that it'll do the work for you. And the app will do all kinds of nice things. So the app will help you to design the experiment, and it will make work lists. So if you've got a liquid handler, you don't have to manually go with a pipetter and make all these pooled mixtures. It will do it for you. And so one of the things we did with this tool as a demonstration for the paper where we reported it is we went back to that initial experiment that I explained, where we took almost 1,000 samples and we did screening and we did mass spectrometry. And in the original experiment, it took 444 hours of machine time to get the raw data to do the analysis. And we repeated that same experiment and recapitulated the results using just 20 hours and a much smaller set of samples. So my last thing to point out is that this was all the work of two members of the lab, Michael Recchia, who is a graduate student, and Tim Baumeister, who is a postdoc. Michael has a poster here this week. He's here in the audience over there. And so if you're interested to hear more about multiplex MS and you want to hear the latest and greatest stuff that's coming out of that project, I do encourage you to go and pay him a visit. I'm sure he'd be very welcome for the company. If you want the tool or any of its technologies, you can get that here. So here's the link to the app, the code base, the documentation, and the paper if you're interested in those things. And then, lastly, I guess I hope I've been able to sort of demonstrate how experimental scientists and informaticians working together are maybe changing the way that natural products as a field starts to think about some of the challenges that we face, and how some of these tools and technologies are maybe contributing to some of those efforts. Lastly, of course, I always have to thank the students who do all the work, the funders who provide the resources to help, and then Waters, who've been fantastic collaborators over many years now, for whose support we are definitely most grateful. So thank you all very much indeed.